Hello, my name is Fábio Malcher Miranda. I'm a PhD student in bioinformatics and I work for the Hasso-Plattner Institute. Today, I would like to talk about HiTaC, a hierarchical machine learning model for accurate taxonomic classification of fungal ITS sequences. ITS is a fungal marker gene widely used in taxonomy and molecular phylogeny due to the fact that it contains hypervariable regions flanked by conserved regions, which can be used to amplify and sequence the variable regions. So what differs HiTaC from previous machine learning models for ITS classification? In HiTaC, a local classifier is trained for each parent node of the taxonomic tree. By contrast, in previous machine learning models, there is usually only a single model trained with the leaf nodes of the taxonomic tree. HiTaC's algorithm starts by extracting the ECCB frequencies from both training and test files. This parameter is flexible and can be changed by the user. Next, HiTaC trains a logistic regression classifier for each parent node in the taxonomic tree. After training, HiTaC can make predictions for the test data, following the taxonomic tree structure. Lastly, HiTaC's algorithm finishes by reporting the predictions. In order to compare HiTaC with similar software, the benchmark TAXXI was employed to allow a quick comparison between HiTaC and 17 taxonomic classifiers. Some of these classifiers are homology-based and use BLAST in the backend, while others are machine learn-based and employ algorithms such as KNN and multinomial naive Bayes. Hence, this is a very diverse benchmark. Another advantage of employing this pipeline is that it already has 5 different ITS datasets ready to use. These datasets are modeled based on cross-validation by identity, ranging from 90% identity to 100% identity between the test and training datasets. The datasets with 90 and 95% identity contain sequences with a mix between novel and non-taxa at the genus and species level, which makes them more challenging. Another important aspect to mention about these datasets is that they are very imbalanced. Here in this plot, which is in log scale, we can see that the phyla Ascomycota and Basidiomycota are much more abundant than all the others for the dataset with 90% identity. Similar patterns can be observed in the other datasets. The TAXXI benchmark also contains scripts to compute some evaluation metrics that are a bit different from the ones we usually see in machine learning papers. In these metrics, novel and non-sequences are taken into account, as well as overclassification and underclassification errors. Overclassification errors measure if the algorithms are classifying sequences that are novel and don't have labels in the training data. Hence, they should not be classified at the lowest taxonomic levels. On the other hand, underclassification errors measure if the algorithms are not classifying sequences that do have labels in the training data. Ideally, both under and overclassification errors should be low. For the results achieved, we can see in this plot that overall HiTaC has a higher accuracy than all the other methods for the dataset with 90% identity. For the remaining datasets, HiTaC still has slightly better accuracy or ties with top methods. For example, in the dataset with 100% identity, it achieves a perfect score along with btop, ktop, microclass, and top. For the true positive rate, a similar pattern can be observed. HiTaC either improves or ties with top methods in all datasets. As for the misclassification rate, lowest values are better. Here, HiTaC has a median score. It doesn't have the lowest misclassification rate nor the highest, putting it somewhat in the average. Remembering that we also strive for a low underclassification rate, HiTaC achieves 0 underclassification rate since its current algorithm always tries to classify all taxonomic ranks. However, by contrast, HiTaC along with btop, ktop, microclass, and top score 100% overclassification rate. Again, we would like the overclassification rate to be as close to 0 as possible. This behavior can be explained by HiTaC's current algorithm, which always tries to classify all the taxonomic ranks, even when they have a low prediction probability. In future releases, we are planning to improve the computation of prediction probabilities and fix this issue. Extending upon the TAXXI benchmark, we performed a resources benchmark to assess whether HiTaC's algorithm is fast enough when compared with state-of-the-art algorithms. For the dataset with 90% identity, we can see that HiTaC takes about 3 minutes and a maximum of 14 gigabytes of RAM to complete. Methods that run in seconds are implemented in C or C++, while the slower methods are either homology-based or implemented in interpreted languages like Python, which is the case for HiTaC. One advantage of having HiTaC implemented in Python is that it's developed as a Chime2 plugin, so it can easily be integrated into existing pipelines. It's possible to easily install HiTaC with pip or conda, and there is also a Docker container available. In summary, HiTaC is a hierarchical machine learning model for accurate fungal ITS classification. HiTaC is able to handle imbalanced datasets and outperforms state-of-the-art classification methods on ITS datasets available in the TAXXI Benchmark, consistently achieving higher accuracy and sensitivity across different taxonomic ranks, especially on lower taxonomic levels such as genus and species.